So, continuing the cell structures, just to show you a little bit of cytoskeleton. Here's a drawing of it showing microtubules, microfilaments, and not entirely sure what the red thing is supposed to be. It doesn't seem to be particularly labeled anywhere. This actually looks like a system of intermediate filaments. So anyway, this is probably a freeze fracture picture done with a scanning electron microscope. This is the nucleus. This is some of the stuff, the matrix holding the nucleus in place. Gets to be a bit of a mess in there. Here's a drawing taken from electron microscope investigation into, so you get these bands, you get basal bodies that produce the microtubules that run up. This would be 2 rows of cilia. A lot of mitochondria. Cilia are very energetic. You need something cranking out a lot of ATP. Let's see. Mucocysts. Is that there's some specialized stuff on here. This is from a parasitology book, or not a protozoa book, where they show some stuff. But you can see that there's connections. It gets very complex in there. It really is very skeletal. So we're going to talk about projection structures. Structures that are contained in projections of the cell membrane. Is that here you've got flagella and cilia. He told you that? Well, he's pulling your flagellum, Nancy. Pseudopods. This is sort of a pseudopod, sort of. Not really. Pseudopods, so you take a cell and you put it on the surface, and it makes a fairly thick projection in a direction. And then you start to see flow into the projection, and basically the projection gets bigger and the cell behind gets smaller, and eventually the cell's over here. And then maybe you get a projection in this direction. You get flow going into that. And if that's the way it wants to go, eventually the cell is primarily over there. So you watch this, and it's kind of like you're looking through the microscope going, so what's going on here? And you go, well, they're just kind of crawling along. Is that's like, like they're putting a foot out and kind of moving it onto the foot. But it's not really a foot, because feet don't really work that way. What are we going to call it? Well, this, remember, is old school microscopy. This is 100, 200 years ago. So they decided they were going to call it a fake foot. And that's what a pseudopod is in Latin. So these are cell projection structures. Probably heard that story from the first semester. Pseudopods are very much driven by actin filaments, microfilaments of actin. They squeeze the membrane and are involved in driving the flow that goes in that particular direction. They also do something kind of neat in that these things are light and they're in water. So they don't when I put my foot down and move, it's friction between my foot and the floor that primarily allows me to do that. Whereas these guys are too light and buoyant to do that. When a pseudopod goes out and starts to pull stuff back in behind it, it needs to stick there. and actin filaments through the membrane adhere to the surface in order to allow that to happen. If you take an amoeba and you put it on a surface where actin cannot adhere, Teflon will do that. What you see is they put a pseudopod out, and as soon as they try to flow into it, it just comes back because it's not stuck down. It's interesting, they get frustrated. Pseudopods can be very thick projections. They can be ornate projections. They can actually be nets. Some protozoans use pseudopods as nets to capture stuff. They can be radiolarians, have these long, thin ones that I mentioned in the last video, and that helps them to float. They look like sunbursts is where the name comes from there. And some of them actually use them as tentacles to snare food objects. So they can be thick, thin, everything in between. They tend to have cytoplasm inside them. These next two do not. I'm going to give you a list. Again, we had this list in the first semester. To differentiate flagella from cilia, but they are homologous. They are built from the same basic internal structure. There's two packets of microtubules in the middle and a ring of 9 around them. And that isn't 100% universal, but it is incredibly close to universal, that 9+2 arrangement in flagella and cilia. But beyond that, they're fairly different from one another. So here's a nice flagella tend to be bigger, cilia tend to be smaller. Again, in biology, you would think there'd be a threshold, like, okay, if it's bigger than that, it's flagellum, if it's lower than that, it's cilia, but they really look at some of these other parameters to decide whether they're looking at flagella or cilia. But typically, I mean, there is such a thing as big cilia and such a thing as small flagella, but by and large, that's a bad pun. Flagella tend to be not just larger, but significantly larger than cilia. It is highly unusual to find more than 12 flagella on a cell. There are some rare protozoans that live inside wood-eating insects that have many flagella, but they're the exception to the rule. Typically, it's 1 or 2 or 4 or 6. They often are paired. It's very difficult to find any type of cell that has more than 12. I don't know that it's possible to find a ciliated cell that has 12 or fewer, because usually there's hundreds of cilia. It's common for the cells to be covered in cilia, but if they're not covered in cilia, they have bands of cilia, where again, there's hundreds and hundreds and hundreds of them. So they're very numerous. On whatever cells you find cilia on, there's very numerous. Since flagella are bigger, you can find them modified in various ways, where they have combs on the end or attached to membranes, so you can make a fin out of them. There's sticky spots on the end. There's a whole bunch of ways you can add on to a flagellum. It's a big enough structure you can mess with it and do different things with it. Cilia are small and simpler. And the closest you get to being able to do that with cilia is to take multiple cilia and combine them into a single structure. That's not really an add-on, that's a merger. And even that's fairly unusual in cilia. And it's not true for all flagella, but it is, if you're looking for add-ons, you're gonna find them in flagella. when you've got a whole bunch of these things moving on a cell, number one, you don't want them to tangle and get in each other's way. Number two, you're generally producing a wave of motion, and you need to generate a wave, so you need a coordinated pattern of push and retraction. So cilia tend to be extremely coordinated, and flagella, when there are multiple ones on a cell, don't tend to, they work together somewhat, but they're not coordinated. You don't see this type of adjustability and coordination with most multiple flagellated cells as you do with cilia. This next one isn't that dependable, but it's useful to know. These things move in a lot of different ways, but a typical flagellar movement is a spin, like it works like a propeller. A typical ciliated movement is a stroke, like a swimmer stroke, where you push and recover and push and recover. And those are the basic differences between flagella and cilia. There's another type of cell projection. So this is something else. Microvilli, little hairs, literally, that's what it means. These look like cilia, but they don't have a microtubule core, they have a microfilament core. Their movement abilities are much more, are much less, is that they can wiggle. And they are primarily an extension of the cell that takes a flat surface and increases the surface area. And being able to wiggle allows materials to get down into the gaps and come in through the edges of this. Right? If they were very static, then this would become very stagnant areas and you wouldn't get as much motion through this part of the surface as you get across like the top. So it would work almost like it was just a flat surface. But with wiggling, it does, so you find these in cells that are moving stuff through the membrane. Maybe it's moving in, like you'd have in the lining of a digestive system. Maybe it's moving out, as you have in some of what they call the brush borders of tubules in a kidney. And very often they are on a surface projecting into a space on an epithelial surface. And because of the way they look through an electron microscope, they're very often called a brush border. It just looks like, you know, they can be pretty tight. Okay, I'm squeezing this in here because I don't want to waste too much of this clear stuff that I write on. So I'm squeezing this in. You may want to give yourself more room. I'm going to write it and then we talked about how one of the functions of carbohydrates is to kind of attach as prosthetic groups onto surface proteins and be a way to differentiate markers in a multi-celled system. Those structures are called glycocalyx. And glycocalyx are what makes the surface of a cell different from another cell, from another person, or from another species, even though it's a cell for the same basic purpose. So we have different glycocalyx, say, on our respiratory surfaces than a dog would have, and I have a different one than probably you have, just different enough that it makes transplanting difficult, because it's part of what your immune system's tasting to decide what's you and what's not you. There's a bunch of ways that cells are held together in a multi-celled system, and those are junctions. They can be tight, zipped together, they can be loosely attached to one another, they can attach in a way where they actually share materials between the cytoplasm in two different cells. Think of all the different ways you might want to attach cells together, and you'll find molecular complexes that do that. And of course, we've talked about cell walls before, where they're a casing around the cell membrane, found in Monera, found in Archaea, found in some protists, found in plants, found in funguses. The materials vary, but the casings are all over the place. Animals, really, the only kingdom that you do not find cell walls. Not all protists have them, so it's not a characteristic of protists, but it's characteristic of everything else. It's characteristic of all fungus, all plants, the two different types of bacteria. And that is it for this chapter on cellular structures. Throw a couple of things up here. There's the 9+2 arrangement, 2 microtubules, 2 little packets, or 9 packets going up a flagellum in this case, but it would also be going up a cilium. Hello, my name is, these are pseudopods. And I think that's all I've got good pictures of.